third plenary speaker of today, Professor Julius GUCCIONE. Julius GUCCIONE is Professor of Surgery, the Surgery Division of Adult Cardiothoracic Surgery at the University of California, San Francisco, United States. And his research focuses on the realistic simulation of the efficacy of novel surgical procedures and devices for treating ischemic cardiomyopathy. Most of you will know for sure Professor GUCCIONE for his contribution to biomechanics, where the GUCCIONE law, I would say, is only the tip of the iceberg. For his manifold contributions, the American Institute for Medical and Biological Engineering inducted Dr. GUCCIONE to his College of Fellows in March 19, and the election to this College of Fellows is among the highest professional distinctions according to medical and biological engineers. So today, Professor GUCCIONE will talk about precision medicine in human heart modeling. So please, Professor GUCCIONE, the stage is yours.
Thank you, Andrea, for that nice introduction. Can you hear me okay?
Yes.
Great. So like Professor Trianova, I'm going to give an overview presentation and talk about 4 projects. I don't know if you looked at my abstract, but it's a pleasure for me to give this plenary session, and I thank the organizers and committee members for the invitation. The title of my talk is Precision Medicine and Human Heart Modeling, which is also the title of a recent paper reference here at the bottom, Pierlink et al. And it talks about precision medicine and also motivation for an in silico clinical trial, a joint venture between Dassault Systems and the United States Food and Drug Administration. And the focus is on functional mitral regurgitation and edge-to-edge mitral valve repair. So that'll be one of the 4 projects I'll provide an overview for. The next one is heart failure and a novel treatment that turns out to prevent adverse remodeling, further dilation of failing hearts. Then I'll talk about a whole heart model that's used to simulate the coupling between the left ventricle and the aorta to provide some insight into the severity of aortic valve stenosis and deciding on whether a replacement is needed. And the last project I'll provide an overview for is a novel approach to closing off the left atrial appendage, which is a site for thrombosis and a potential stroke for patients suffering from atrial fibrillation. So after I provide an overview of those 4 projects, I will I have a couple summary slides pointing out the most important points. And then finally, a last slide before the acknowledgement slide, where I talk about future directions for these 4 projects. So there are basically 2 types or classifications of mitral regurgitation. There's degenerative and functional. Degenerative could be mitral valve prolapse, where you could have a chordae rupture. So there's something abnormal about the valve. In functional mitral regurgitation, there's really nothing wrong with the valve, but you have backward flow, which I'll show in the next 2 slides, some movies of mitral regurgitation due to changes in the left ventricle. And one thing I want to point out with this slide is how common mitral regurgitation is for patients over the age of 75, and also that about half of those patients are really not good candidates for open heart surgery, and that's why percutaneous edge-to-edge repair like the MitraClip, marketed by the company Abbott, or the Pascal device by Edwards Lifescience, are important to the FDA and this in silico clinical trial that they're working on. And if the mitral regurgitation is not addressed, this will lead to heart failure. So this first movie shows a whole human heart model. It's a normal heart, a 50-year-old man, and it's an abacus simulation. And the reason for the mitral regurgitation, these little spherical particles that show up in the left atrium while the left ventricle is ejecting, is due to chordae rupture. So this is prolapse and degenerative mitral regurgitation. The next movie is functional mitral regurgitation. Again, there's nothing wrong with the valve, but the annulus of the left ventricle has dilated. So we have a little bit of mitral regurgitation here in the left atria. So there are different approaches to mitral valve repair. Even the cardiac surgeons will agree that mitral valve repair is preferable to mitral valve replacement. And again, because of the prevalence of mitral regurgitation in elderly patients, and they're not good candidates for open heart surgery, the percutaneous approaches like the MitraClip are gaining attention. Now, I apologize, this slide doesn't fit very well, or the information doesn't fit very well on this slide, but I just want to point out a couple things. The EV stands for Emiliano. Here's Emiliano's name, Vata. He works with Alberto Redielli in the biomechanics group at the Politecnico di Milano. They're mitral valve experts, and Emiliano is an expert in the finite element software Abacus. So Emiliano shared his Abacus model of the mitral valve with Dassault Systems, and they've been working to adjust the whole human heart model so that the mitral valve behaves as well as Emiliano's Abacus model. And you can see in this slide that this is some information about version 1 of the Dassault Systems model. And they've made a lot of progress and have managed to match the behavior pretty well of Emiliano's state-of-the-art model. And I'll show this movie, a side-by-side comparison. And so thanks to Emiliano and Alberto, their willingness to share the in silico clinical trial is off to a very good start so far. So there are a lot of advisors for this enrichment trial, and they decided that rather than use a whole heart model, a left heart model that ignores the right ventricle and the right atria is a lot easier to deal with. So this movie shows a Abacus model of the left ventricle and the mitral valve. This next movie shows just the mitral valve apparatus, including the anterior and posterior papillary muscles. So you can appreciate the importance of including the left ventricle so you know the motion of the papillary muscles. And then this movie shows the kind of an overview of the left heart model, again, looking at cross sections, the mitral valve, papillary muscles, the mitral valve apparatus, and the whole heart model. So this is a brief summary of the progress so far. And I guess I consult for Dassault Systems, and I'm part of this scientific advisory committee for the enrichment trial. And I guess my input right now is basically getting them to validate their left ventricular model by making comparisons with clinical measurements of stress and strain in different regions of the left ventricle. Okay, now that may seem like a kind of an abrupt stop for that project. The next step is going to be making the model patient-specific, and I will provide some slides on how to make models patient-specific, but I'll do that in my overview of the third project on aortic stenosis. So now we're on the second project concerned with heart failure, which is a worldwide epidemic, affects 26 million people worldwide. One of the reasons why it's so prevalent is the cardiologists are getting very good at treating myocardial infarctions, getting patients, getting the blocked coronary artery open within 30 minutes. But then after the patient survives, a number of the patients develop heart failure. Now I've been doing some research to try to figure out ways to improve this novel treatment that involves injection of a hydrogel, and we know it's not perfect, so we're considering combining hydrogel injection as a treatment for heart failure with reduced ejection fraction in combination with vagus nerve stimulation. And it's quite interesting that in heart failure, there's an abnormal increase in sympathetic tone. There's a decrease in the compensatory activities of both the parasympathetic nervous system within the neurocardiac reflex hierarchy and the muscarinic systems that affect the cardiomyocytes. These changes increase oxidative stress, impair calcium signaling, and adversely alter cardiomyocyte gene expression. These changes are arrhythmogenic, for example, atrial fibrillation, and pro-ischemic, meaning that you could suffer another heart attack. And vagus nerve stimulation, abbreviated VNS, can mitigate or reverse these adverse effects, but it doesn't seem to be a cure on its own for heart failure with reduced ejection fraction. So that's why we're proposing a combined treatment of polymer injection to reduce dilation and vagus nerve stimulation to improve strength of contraction or contractility. Now, another point about heart failure is a cornerstone in treatment of heart failure is reducing the stress on the muscle fibers, the load on the muscle fibers. A failing heart with reduced ejection fraction is typically twice as large as a normal heart. So with Laplace's law, you would expect the stresses to be abnormally high. And we obtained MRI data from 3 patients treated in Munich, Germany, with the polymer injection in addition to coronary artery bypass grafting. And the results were amazing. There was a sustained decrease in left ventricular volume at the end of filling, abbreviated ED for end diastole, and especially at the end of ejection, end systolic volume was cut in half at least. And this is remarkable, and you wouldn't expect the coronary artery bypass grafting to produce this effect. But a major question is, what effect does the hydrogel injection alone have? But this combined treatment reduced stress in the local muscle fiber direction by at least 35%, both at the end of filling and the end of ejection. So we got a grant from the National Institutes of Health to create heart failure with reduced ejection fraction in swine, and then do computational modeling to see the effect of polymer injection alone, and to try and understand the mechanism of action. So we studied both, we studied swine before creating an infarction and afterwards, and heart, like atherosclerotic plaque rupturing, the myocardial infarctions were produced percutaneously. And it took at least 8 weeks for the ejection fraction to drop down to 35% or less. And then 8 weeks after myocardial infarction, that's when the polymer injection therapy was performed. And we also had at least 5 animals where we followed them out for 16 weeks, like the heart failure injection group, as a heart failure control, HFC. And we also did detailed computational modeling on normal swine, normal controls, which we abbreviate as NC. So here are the finite element meshes created by Kevin Sack, and it's all the details are provided in this freely available paper. And you can see visually the injection pattern. There are 12 to 14 injections in each heart, only in the free wall, because this was done with an open heart injection protocol. And there are 2 circumferential rows. And again, the goal was to do detailed modeling using ex vivo diffusion tensor magnetic resonance imaging to build very detailed models geometrically, anatomically, and to try and understand the mechanism of action. So this slide shows an ex vivo left and right ventricle from a swine heart, and in blue we have the large myocardial infarctions, multiple infarctions. And then we know that the contractility, even in the border zone, which is shown here in green, so it's normally perfused, it's adjacent to the myocardial infarction, but the active stress that's developed by these muscle fibers is only half normal. So Kevin introduced a parameter he calls a health index, and provided a continuous variation in the strength of contraction depending on the infarction, how many muscle fibers are remaining, and how far the tissue is from the infarcted region. And then here we can see a untreated heart failure 16 weeks after myocardial infarction and no polymer injection. And then we see another swine left ventricle that was treated with polymer injection. You can see the gray blobs here. This is alginate mixed with calcium, and it causes the infarcts to be much smaller. And I'll show in a movie that the motion is much better in panel F versus panel D. In collaboration with Dan Ennis and Eric Aliotta, when they were at UCLA, now they're at Stanford. They did overnight ex vivo DT MRI scanning to get very detailed measurements of muscle fiber orientation in the left and right ventricles. And here you can see the typical pattern where the muscle fibers are circumferentially oriented at mid-wall, and then perhaps a plus 60 degrees with respect to circumferential at the endocardium, minus 60 degrees with respect to circumferential at the epicardium. And we also take into account the papillary muscles and the unidirectional muscle fiber orientation towards the mitral valves. And then, as has been mentioned in many talks, great talks that I listened to earlier today, we have a 3D finite element model for the left and right ventricles, but then 0D models keeping track of the blood flow between the different chambers. So this movie is a side-by-side comparison of the cardiac cycle for the untreated heart failure control on the left with very little wall thickening. And then on the right, we have polymer injection. The polymer is removed here, but the size of the infarct is reduced, and we still have some variation in wall thickness throughout the cardiac cycle. Now, this slide is very helpful. It shows the pressure volume loop, but it's normalized for end diastolic volume. And here we have the untreated heart failure with the smallest stroke volume. And then here's the normal heart failure control, NC. And then the treated heart failure is kind of a compromise or intermediate. So the polymer injection provides some protection so that the contractility of the untreated heart failure is not as depressed. The treated heart failure is not as depressed. The stroke volume and injection fractions are increased, but it doesn't cure the heart failure with reduced ejection fraction. It just provides some protection. So we also found that when we looked at the unloaded hearts, the untreated heart failure left ventricular volume was 45% greater than normal. But in the treated hearts, left ventricular volume is only 4% greater than normal. So that's why we call it a left ventricular mid-wall constraint mechanism of action. And passively, the untreated heart failure is stiffer, the infarcts are stiff. And actively, again, you can see that the muscle fibers don't contract as strongly as in the treated heart failure and certainly the normal swine. And interestingly, the polymer injection seems to help the diastolic function and a reduction in end diastolic pressure. Now, in collaboration with collaborators with expertise in electrophysiology, we've used the same detailed models to predict activation times and action potential duration and comparison between normal controls, heart failure controls, and the injected treated heart failure, where the gels are indicated here and the infarcts here. And the take-home message is that the gels do not adversely affect ventricular repolarization. So it's not going to be pro-rhythmogenic. Okay, moving on to the third project overview with aortic stenosis. And this is work done by a surgery resident at UCSF, Andrew Wisniewski. And the work is available in this freely available paper. Aortic stenosis is a prevalent aortic disease, heart disease, valvular disease. And if it's not treated, again, you could end up with heart failure. Now, the way that the severity of aortic stenosis is assessed clinically is 3 ways right now: transvalvular pressure gradient, effective valve orifice area, and ejection blood jet velocity. So really focusing on the aortic valve. But in the case where there's low flow and low gradient aortic stenosis, this type of grading has limitations. So Andrew's using a whole heart model, whole human heart model, and adjusting the resistance in the 1D model of the aortic flow to cause, here we have the normal left ventricle, moderate aortic stenosis, and severe aortic stenosis. And this is pressure volume loops. And on the right, we have pressure versus time. And the dashed lines or dash curves are the pressures inside the aorta. And the difference between left ventricular pressure and aortic pressure is the gradient. So one thing that the clinicians are not using, because it's not easy or perhaps impossible to measure, are the myofiber stress distributions shown for the three cases at the end of diastole and at peak systole. And the stress in the local muscle fiber direction is color-coded with the highest tensile stress in red and the lowest compressive stresses in blue. And there are significant differences in peak systolic stress between these 3 cases. And here we can see stress in the local muscle fiber digression, peak systolic stress for the 3 cases. But this is for the American Heart Association segments 1 through 17, where 1 through 6 is the basal region of left ventricle, 7 through 12 is mid-ventricle, 13 through 16 is apical, and then 17 is right at the apex. So you can see the greatest differences in stress between severe aortic stenosis and normal is this is the basal inferior region. This is the anterior septal region, mid-ventricle, and then right at the apex. Now, what we're doing, work in progress, is to make this left heart, start focusing on a left heart model and making it patient-specific using CT data from a cardiologist at UCSF, Vyakam Mahadevan, and Salvatore Pasta, Salvatore Coutuno in Palermo were really helpful in helping us digitize the CT data. And here you can see the model of the left ventricle and the left atria of the aorta based on the CT imaging. And then we pass this information off to Zhang Yao, who's the technical lead at Dassault Systems for their Living Heart project, and Ashley Stroh, another engineer at Dassault Systems. And in addition to Abacus, they use really state-of-the-art computer-aided design software called CATIA to morph the normal human left ventricle geometric model and have it adapt the geometry of the patient-specific CT data. And this is also being pursued, patient-specific modeling for the in silico clinical trial involving functional mitral regurgitation and edge-to-edge mitral valve repair. All right, the last project involves a novel approach to closure of the left atrial appendage. And here's a figure off the internet with a device that is used to close off the left atrial appendage so that there aren't any blood clots and a stroke due to atrial fibrillation. But my colleague at the California Medical Innovations Institute in San Diego, Ghassan Kassab, came up with a novel approach that doesn't leave any device behind. So percutaneously, they have a catheter that grabs the inside of the far end of the left atrial appendage and inverts it. And then, although it doesn't look like anything's changing in panels EF and G, they actually, with the same catheter, will use surgical glue so that the this inverted left atrial appendage stays inverted. And then here they've removed the catheter. So with Zhang Yao, she helped use the left atria from the Dassault System's whole human heart model to simulate inversion. So here's before inversion and after inversion of the left atrial appendage. And then Salvatore at University of Palermo helped us look at the stress distribution in the left atrial appendage for different degrees of displacement of the far end of the left atrial appendage. And you can see, again, color-coded where blue shows compressive stress. So the idea is that we'll have a combination of tensile and compressive stress that will cause the left atrial appendage to shrink and kind of close off on its own without leaving any device behind, which might migrate through the cardiovascular tissue. That's one of the disadvantages of leaving devices in the patient. Okay, so the take-home messages: the US FDA and Dassault Systems are collaborating on an enrichment in silico clinical trial that focuses on functional mitral regurgitation and edge-to-edge mitral valve repair as a treatment. The mitral valve in this left ventricle mitral valve model is based on the pioneering work by Alberto Redielli, Emiliano Vatta, and co-workers at Politecnico di Milano. For the polymer injection, functional output for the treated heart failure is better than untreated heart failure, but not as good as the normal case, at least in swine. And we believe vagus nerve stimulation may be what's missing in order to cure heart failure with reduced ejection fraction. And then fortunately, the results of Alicio Gizzi in Rome and Daniel Hurtado in Santiago, Chile, it appears that the hydrogel injection does not affect the regional dispersion of repolarization, at least when comparing the treated heart failure versus heart failure control. For aortic stenosis, peak systolic stress averaged 9.31 kilopascal for baseline, 13.13 kilopascal for moderate aortic stenosis, 16.18 for severe aortic stenosis. Peak stress was significantly elevated compared to baseline for moderate and severe aortic stenosis. The p-value was smaller for severe, and it was also significant when moderate stresses for moderate aortic stenosis was compared to severe aortic stenosis. Ventricular regions that experienced the greatest systolic stresses were the basal inferior region, mid-anterior septal region and the apex. Using a realistic left atrial anatomy as extracted from the Dassault Systèmes living heart human model, the left atrial appendage tip was virtually clamped and the retraction procedure was simulated to reveal insights on biomechanics of inverted left atrial tissue wall. The simulation confirmed the fundamental hypothesis of our study, suggesting a change in the stress distribution of the left atrial appendage inversion tissue wall from tensile to compressive as a result of the inversion procedure. The next steps for the inversion enrichment trial are to make the LV mitral valve models patient-specific and to accurately simulate clinical endpoints, mortality and rehospitalization, typical clinical endpoints in clinical trials. The next step for the left ventricular mid-wall constraint treatment of heart failure with reduced ejection fraction is to add vagus nerve stimulation and then, if successful, perform clinical trials. The next steps for the left heart modeling of aortic stenosis are basically the same as the ones above for the enrichment trial. The next step for the left atrial appendage inversion study is to repeat experiments in swine that have atrial fibrillation. There have already been experiments performed in normal swine that show that the inversion of left atrial appendage does cause resorption of the appendage. Now, as mentioned in previous talks, the Achilles heel of these detailed abacus models is they take like 3 days to run, so that finite element surrogates, reduced order models are useful in getting simulations and reproducing output from the finite element models in a matter of seconds, ideally, as opposed to 3 days. And if you have these finite element surrogates and train machine learning algorithms, this would enable real-time prediction of patients' specific MitraClip device outcomes. That's something that is also an ongoing project with Yagub Dabiri at GUCCIONE's Institute. And this is my acknowledgement slide with the many people that collaborated on these 4 projects. And thank you for your attention.
Thank you, Professor GUCCIONE, for your very nice talk. The floor is open for questions, so unmute yourself or raise your hands if you have questions. Perhaps I can start by asking you how the say computational pipelines you have showed in order to target the applications you have talked about can be tailored somehow to a new patient. So which is the effort required to tailor, say, all the procedures you have shown to take into account the new case, so in order to personalize the model once again?
Yeah, so that's a great question. Ideally, you would get the type of medical imaging data that Professor Trinova described in her coffee, answering the excellent questions that were asked during that session. There are limitations with the different pluses and minuses with the different imaging modalities. And I think one of the most important things, as pointed out by Luca Dedi in his plenary talk this morning, muscle fiber orientation, and I've heard presenters talk about rule-based methods. I think that's why we think the swine experiments are so important We have ground truth for muscle fiber orientation. And I think if I had a lab as large as Professor Corteroni, I would ask students to do comparisons of results from the swine model, where we have ground truth for muscle fiber orientation, left ventricular geometry, including the trabeculations on the endocardial surface, and precise measurements of the infarcts and ischemic zones, and then see how this affects your results and conclusions when you swap in a geometry based on patient-specific MRI or CT or echocardiography. But not only are these very sophisticated models very time-consuming to run, but there's another bottleneck in digitizing the medical imaging and building the models from scratch. That's why we get help from Salvatore in Palermo, and also Dassault Systems, which has a vested interest in clinical trials, in trying to save time by, rather than building models from scratch from medical imaging, just use a few control points to morph the finite element or abacus mesh using CATIA to rapidly change the geometry of the model, and ideally start studying patient populations. I hope that answers your question.
Yeah, thank you. I see the answer of Professor Trajanova.
Hi, thank you very much. It was really such a pleasure to listen to you. I want to ask you a question about your studies where you did high resolution diffusion tensor mapping of the fiber orientation in these infarcted pigs. So we did the same. It was a few years ago when Elliot McVeigh was here. So we scanned a number of pig hearts with infarction at the NIH scanner, like overnight and stuff. And we did a very extensive study on the fiber orientation. We have 2 papers published on that one, just on the fiber orientation and the other simulations with these pig infarcted models. But I wanted to ask you, one question we always have been asked when we construct these models is, and there is an infarction, everybody goes, oh, we think that there is a potentially fiber disorientation in the infarct. And our study showed that there isn't. Basically what happens is, while the cells die, the original tracts remain. So the water diffusion is along those tracts. There is no like chaotic fiber or water diffusion tract lines in the infarct. So we measured that, and we just found that because the tissue is thinner across the infarct, is just the faster change in the fiber orientation. But per unit length, the change in fiber orientation was pretty much the same across the wall. What's your impression from your studies?
Well, thank you, Natalia. I had not heard about that source of discrepancy or artifact in trying to measure muscle fiber orientation in infarcted hearts with diffusion tensor MRI. So thank you for bringing that to my attention. And I did read your paper with Elliot, and I love the title of that paper, pointing out the sub-millimeter spatial resolution. So we also published a paper fairly recently in kind of an obscure journal. I think it's a material science journal. And without taking that artifact into account, we did notice some changes or differences in muscle fiber orientation between infarcted hearts and normal swine, and in certain regions. But one thing that we really wanted to know is what effect the polymer injection has on the muscle fiber orientation, because if it has a bad effect, then that's going to make things worse. Right. So our real goal was comparing what is the effect of the polymer injection on muscle fiber orientation, how large is it? And we found that it's not as large as what we thought was the effect of the infarction. But yeah, I have to go back, and we have that that data, and perhaps we'll have to correct that that assessment of the effect of ischemia and myocardial infarction on muscle fiber orientation. So thank you very much for your question.
Thank you very, very much. Thank you.
My pleasure.
Hossein?
Yes. Thank you, Dr. GUCCIONE. It was a really great talk. So I really enjoyed. So basically, maybe I misunderstood, but I was wondering, were your models able to capture the growth and remodeling in response to these two types of valvular disorders? And if yes, what was the driving signal in your models?
That's a great question. And again, I'm going to bring up some nice answers by Natalia during her coffee with the students. I think it's so important to realize that the models should be problem dependent. As Natalia said, there's no model is going to provide answers to everything. So to answer your question really quickly, there's no growth modeling. I think that may be one of the most difficult challenges we have. It's important. I noticed that one of my former students, Jonathan Wank, is a co-author on an abstract in this meeting. So I defer you to their growth modeling. The abstract sounds like they have really broken some ground with a unified growth model that can handle different types of hypertrophy. But to answer your question, there wasn't any growth modeling in any of the four projects that I pointed out. Of course, growth modeling is hugely important to understanding the left atrial appendage inversion and determining the mechanism of action and seeing if it's compressive, changing some of the tensile stress into compressive stress after you invert the left atrial appendage to understand the mechanism of action. That's probably all I should say in answer to your question, but that's a great question. Thank you.
Thank you very much. So actually, I'm Jonathan's student, so thank you very much.
Carla. Apparently she disappeared, or well, if there are no further questions, we planned in the program.
I forgot to unmute.
It is Carla. Yeah, of course.
Yes, I was trying to say if you could please go back a couple of slides.
What am I looking for?
I'm trying to remember. I'm sorry. Could you slow down a little bit, please? Okay. Yes. So the first question is very, very general. So I'm sorry. Why are we still modeling? Why are we still using pig models?
Great question. And I'm going to mention another abstract I saw that included colleagues Christian Stock and Sebastian Kozerki. They're one of the three, they have one of the 3 or 4 groups in the world that can measure muscle fiber orientation in beating hearts, but the spatial resolution is nowhere near what we can obtain in non-beating hearts. And apart from getting the explanted hearts from after a heart transplantation, I don't see any way to get human hearts for ex vivo diffusion tensor MRI. And again, this is going to be very technical, but the constitutive relations that will allow you to produce realistic left ventricular motion in agreement with strain measurements by tagged MRI, dense MRI, or speckle tracking with echocardiography. The success of those constitutive relations and your model will depend greatly on the accuracy of your geometry. If you have a smooth endocardial surface, as most of the models that I've seen so far, you're going to get very different mid-wall strain in comparison with the MRI-based strain measurements, or endocardial strain with the speckle tracking and echocardiography. So I really think.
So a lot of the issues, yeah, sorry, a lot of the issues come from imaging?
Yes, yes. So
okay, well, if that's the case, then.
Go ahead.
Yeah, so if that's the case, then why are we imaging the wrong thing?
Okay, because at least in the United States, everybody is trying to, or most people would like to get an NIH grant, and you have to propose something that is part of the NIH National Institutes of Health mission, which is improving patient care. So that's the goal. The carrot is patient-specific modeling, and now in silico clinical trials, virtual patients, all that. But you can't measure nearly as many things in patients as you can in animals. And I know that the general public is against animal testing, but I think the modeling community can say, look, we're going to make sure that these animal studies, that data is going to be used to the fullest. We're going to create computer models. I love this LifeX effort for open source computational modeling software, so you can share these pig models with other people. So if you have ground truth for geometry, muscle fiber orientation, and ischemic regions and myocardial infarctions, then you can understand the disease that much better, understand the mechanism of action of devices and treatments, and then hopefully we will use patient-specific imaging data to make these pig studies, modeling studies, translational, so they are relevant to doctors. And I didn't point out one clinical application, part of Kevin Sachs' thesis with Tom Franz in Cape Town, South Africa, was left ventricular assist device modeling and effects. So he took everything he learned from the pig studies, got an MRI data set from Scott Solomon at Massachusetts General Hospital, and used that patient-specific biventricular geometry to simulate the effects of left ventricular assist device. And we found that if you turn up the continuous flow LVAD too high, you're actually going to invert the septum so that it's convex instead of concave. So that was very helpful and also translational.
Okay, but yeah, I'm sorry to keep pressing the question. I'll put my hand down in a second. Just one last question.
Okay.
If we're already moving towards translational, sorry, how to say this? Shouldn't the ionic models be good enough by now that we don't keep, like, when are we going to stop using animal models? It seems like it's never really going to stop. Like, it seems like our models should be good enough by now.
Yes, so good question. And I'm glad you brought up ionic models because as I pointed out, I reach out to people that understand electrophysiology better than I do. And again, for these 4 projects, the models were problem specific and there was no ionic modeling. We assumed that the muscle fibers all contract at the same time. And we're really focused on what's happening at the end of ejection and systole or peak systole. So a purely mechanical model, even using simplified active stress constitutive law like time-varying elastins, still provides useful insight and fills some gaps in these 4 disease areas and applications. But please send email to Professor Trianova to get an answer on why the ionic models aren't yet good enough for all the doctors in the world to be using at the same time that they're ablating patients.
I think we should move that meeting. It's not a subject of really, we are holding an entire international meeting with.
Sorry.
Thank you, Julius. I very much appreciate your answers. It's these questions are really not for a 10-minute question and answer session. So I'm sorry for taking the liberty for saying that, but I'm really interested to hear other people's opinions as well.
Yeah.
Thank you.
Thank you.
I think otherwise you can also stop here, since we also plan to have a coffee with Julius at the end of the session of today. I think we can stop here and say goodbye to tomorrow, when we'll have the morning session about the code, and then the afternoon regarding European Central Summertime session with plenaries and contributed talks. And if people are still interested to ask questions to Professor Guccione, they can stay still online in the virtual room to have a virtual, unfortunately only virtual, coffee with him, sharing their thoughts and questions. So see you tomorrow. To all the attendees, we'll reconvene here tomorrow.
Thank you, Andrea.
Thanks to you. Luca, I ask a technical question to Luca Paglieri. The virtual coffee is still here, right? In the same room.
In fact, it's just a formal, let's say,
of course,
transition, but it's okay if you continue talking as long as you want.
Yeah.
In any case, there is only this room active, so everyone interested can stay.
Either we can wait for people, say, leaving the virtual room, or say
no, you can continue without stopping. People can stay what they will do silently.
Okay, so we can continue here. All right. So
Dr. Guccione?
Yes.
How are you? So I'm Hossein. I'm Dr. Jonathan Wang's student. So it's a nice pleasure to see you here. So basically, you know, we have been hearing your name in our lab meetings many times. So it's a really very big pleasure to meet you here. So basically, my question is that how effective would be the precision medicine in heart modeling in the clinical treatment? I don't know, in the near future. For example, how long do you think it would take that these types of modeling, these computational tools would be implemented into the clinical care and in a way to enhance the patient care? Just to ask your opinion or impression.
So I think there are two approaches. One is to form a team, a partnership between a clinician and a engineer that can run the finite element model, and hopefully there's a treatment that is not so time sensitive that you could take up to 3 days to get that finite element solution. And the engineer would run the model and explain the results to the clinician. And hopefully the clinician will have maybe studied math or physics as an undergrad. That would help. So I think, and I trained with Y.C. Fung as a graduate student and read his textbooks even as an undergraduate at another university, Tulane in New Orleans. And he would point out what the orthopedic surgeons, how they treat patients, and they use biomechanics. It's pervasive. It's used throughout their clinical practice for the artificial hips and now artificial knees. So we should look at what the orthopedic surgeons are doing and try to emulate them. Now, so far, it doesn't appear that the finite element method is used in clinical practice. And I think I didn't wake up early enough to see Professor Trianova's talk. I read her abstract and it was very impressive. And I listened to her answer questions during the coffee. I'm assuming that she also is looking at finite element model surrogates. They could be just black box, but be able to reproduce the relationship between the input and the output of the finite element model. And then if you can train a machine learning algorithm, and we've published a couple papers, I can send you the links or the papers via email, where we used machine learning software, XGBoost and Cubist, to not only reproduce the pressure volume loops from a finite element model, but being able to reproduce the stress distribution and strain distribution. So spatially varying metrics at a particular time point in the cardiac cycle. And then for clinical, I think you have a better chance of having a machine learning algorithm being used clinically. It should be much more user-friendly than finite element software for a clinician, especially if they don't have a technical background. And that way you can kind of package the problem-dependent model very concisely, and it should be easy to share. And you don't need a finite element solver, but you need to be careful that the machine learning algorithm is only going to be valid in the realm for which the data exists, the data that was used to train the machine learning algorithm. Does that answer your question?
Yep. Yep. And thank you for the answer. And so I can say that from today's talks, I learned that I think that the machine learning is going to be the key point that kind of connect the computational model with the clinical curve in the, I don't know, near future maybe. But yeah, thank you very much.
Well, just to comment on that, the computational model itself can be used in the clinic. It just depends on the application. I'm going to repeat that many times. We're doing a clinical trial now on ablation, and because the patients are spaced 2 weeks from each other, you can do it. You don't need the machine learning to predict where to ablate. Everything has to be tailored for what is the clinical workflow, what is the best way to slot in there. And I think Julius really sort of emphasized that in his talk, so we're in full agreement about it. I think both are important, but the application is really determines what's used when and how.
Yeah.
Sorry for interjecting.
No, that was perfect. Thank you.
Paolo.
Yes. So thank you for the brilliant lecture. I have a question about in this direction, still in the field of let's say understanding really the translation between the theory, the computational model, and the clinic. And my question is about something that you mentioned at the very end of your lecture. And indeed, let me phrase it like that, is what is the role of the middleman? The middleman for me is this large companies, large computational companies, as, for example, the Sol Systems, as ANSYS, who can really influence the, let's say, the market and the applications and maybe facilitate this translation. So as you mentioned that, just as a curiosity, without breaking any confidentiality, maybe agreement, can you comment that on the possible role of these, let's say, players in this process?
Well, thank you, Paolo. That's was a great question. And I heard you speak earlier today. I think it was you, and you mentioned and perhaps asked a question about, the heart modeling. Will it be applicable to other parts of the body like the lung? That was you?
Yes, just a comment, but yes.
That's great. So, I don't know if you noticed, but my eyes are going all over the place because I have two screens, and one was presenter mode for the PowerPoint presentation, and one is what I have an idea of what you attendees are seeing, and I could tell that I was going too slow. So the one point that I wish I had made is, even before the great interest in machine learning, I would hear from the cardiac surgeons about things that they could measure that are related to mortality. And I think that's really the greatest challenge for an in silico clinical trial, where you're trying to replace some of the real patients with virtual patients, computer simulations, is how do you predict typical clinical trial endpoints like mortality with an ANSYS or Abacus model. And I think because end systolic volume is the one metric that is most closely correlated with mortality, that may be a really good thing to focus on. So, try to get your models to accurately predict end systolic volume and the effect of devices or surgical procedures on end systolic volume. And then I also would have mentioned, if I wasn't racing, that Daniel Hurtado recently signed on with Dassault systems. And I want to point out that they have a very simple non-binding agreement where you're free to publish, and there aren't a lot of restrictions, but there is an agreement in place. Anyway, Daniel Hurtado is one of the principal investigators for Dassault Systems' Living Lung Project. And I really think, especially with COVID-19 being a respiratory problem, that it's important for us to at least include the lungs in our zero-dimension models, and ideally have some finite element 3D models of the lungs that we can couple with the heart, because they really don't work in isolation. So I hope I answered your question, and I was able to include a couple important points that I didn't make during my talk because I was kind of rushing towards the end.
Thank you very much. I really appreciate it.
My pleasure. Thank you.
Is there any further comment or question for Julius? Well, if not, well, maybe we can close this virtual coffee break. And once again, see you tomorrow. Have a nice day, afternoon, evening, depending on the point of the world you are at this moment.
Thank you
so much to our Italian hosts. Thank you.
Very much.
Yes, thank you. You did a wonderful job.
Beautiful talks.
Thank you all. See you tomorrow.
Bye. See you tomorrow.